Okay, the title of my talk is Building a Product with Machine Learning and Genomics, or omics in general, and I will present our one of our products, the NEC Neoantigen Prediction Platform. If you've seen the poster for this seminar, you can see that the title does not align, neither the photo of me is not really up to date. But in my talk, I will try to convince you that having curated, correct data and up-to-date data is very important in developing such products. So let's try a bit of shameless self-presentation. I work at NEC Immunity, which is part of the NEC Corporation. Existed for more than 125 years now. Even though maybe when you hear NEC, you don't think of AI, actually it has produced a large number of papers in top academic conferences. You can see comparing us with the big players here. This AI drives a lot of technology in NEC. What we like to say, from the ocean floor to outer space, so from undersea cables to satellites. And where I come from, NEC Oncoimmunity is part of the NEC Bio group, which is the interface of NEC to the pharma or biotech world. Being part of NEC actually gives us also the chance to be in wider ecosystem where we can collaborate with AI research labs in the US and the EU. And under NEC Bio, we also, of course, it's the NEC Biotherapeutics company in Germany which runs the clinical trials we are part of. That's enough, I would say, about NEC. What are the main research questions that we try to tackle at NEC Oncoimmunity? We are working on designing therapeutic and prophylactic vaccines to tackle both cancer and infectious disease. How do we do this? We try to exploit both arms of the adaptive immune system, the cell-mediated and humoral arm of the adaptive immune system. Currently, we have three products that use machine learning to tackle this problem. As personalized cancer vaccine pipeline, off-the-shelf cancer vaccines, where we try to further develop vaccines that can be used, basically off the shelf to many different patients, as opposed to the first product, and broadly protective infectious disease vaccines. So the same technology used in cancer, we have adapted it to infectious diseases and other pathogens, of course inspired by the pandemic in 2020. And there we try to develop broadly protective vaccines so that cover as much as possible the world population, and as much as possible of the possible threats that come. Today I'll focus mostly on the personalized cancer vaccine product. This is our most advanced product in development, currently part of two clinical trials. So let's see how we got from research, trying to answer a research question, to actually testing it in clinical trials. What it is that we do? So first, every cell in your body, every nucleated cell in your body, constantly produces proteins which do some function in the cell, and then they are cut up, degraded when they're done with their work, and into smaller peptides, which our immune system actually then, there are processes in the cell that take these smaller peptides and take them to the cell surface, where they're presented to immune cells, and this is basically what's called the antigen presentation pathway. With the immune cells then can recognize these peptides as either self or foreign. And when they see something foreign, they initiate this cell-mediated immune response, which then tries to get rid of these cells. There are several processes that are part of the antigen presentation pathway. And we take a holistic approach, trying to model each one of them with machine learning models. Not to get too much into the biology, however, the main processes that we model, HLA binding, for instance, is the process of basically binding of these small peptides to a major histocompatibility complex molecule, which is which has some kind of preference for peptides. The set of peptides that these MHC molecules will bind to actually depends on your HLA profile. This is different for every human. And modeling this process for each patient is what makes this process personalized, this approach personalized. Some other things happen in the cell. These proteins need to be cut up in a certain way, so we try to predict also that. And then the final step is when they are presented at the surface cell, will there be an immune response? Will there be a T-cell receptor that recognizes this peptide MHC complex and initiates an immune response? That's shortly about the biology behind the problem. Now, to implement this, we have implemented an end-to-end personalized cancer vaccine design pipeline. We start with the NGS analysis using the tools that Pubudu already talked about, or at least similar tools, where we take from the patient tumor and normal tissue. The sequencing analysis determines all the variants, all the changes in DNA that can be observed between the normal and the tumor tissue. Also does the HLA typing, so determines what is the HLA profile of the patient, and then generates all the candidates, the small peptides that could possibly be presented at the cell surface. Several models are used, like I already mentioned. This string of beads vaccine type, where each of these peptides that were determined to be best to elicit an immune response are combined. And then, the output of this product is this string of bead vaccine design. How does this work in the clinical trial? So we collaborate, of course, with hospitals and other companies in this process. First, there is sample collection at the hospital. Then this is run through an NGS pipeline to get the sequencing output for both the normal and the tumor tissue. This constitutes the input to our pipeline. Our NGS analysis pipeline, like I described before, processes this data, and our machine learning pipeline produces the predictions. And then the vaccine design is constructed, and this is sent to another company, where this construct is inserted into plasmid or whatever the modularity of the vaccine is, and the vaccine is then administered into the patient. That's part of the clinical trial. That sounds easy, so we developed a product, we put it in clinical trial. But in order to do that, this is subject to regulatory approval. So what does that mean? Well, in order to test your product that affects actual patients and basically we need to prove that this is safe first, and there are certain standards developed by regulatory authorities that you have to comply with. Let's see, what does that mean? The one way to comply with is to implement your own quality management system, which will make sure you follow some standards like good clinical practice, good manufacturing practice, good laboratory practice standards. You have to include here also the standards for privacy, like GDPR or HIPAA, and so on. And you have developed a product with machine learning. That means you wrote some software to run this, and often your product will be categorized as software as a medical device. That brings another set of standards that you need to follow, and then you comply with. I have listed them here. I'm not going to read through all these names, but they are basically make sure that you So what I propose is that, while you develop your product, you follow certain best practices in software development. So the goal here is to develop software that is reliable, scalable, that you can maintain it, extend it, and so on. And of course, these problems you will not solve alone. You need to collaborate in a big team. Always use version control. What does this mean? You need to track all the changes that you do to your software. You implement your first version, you need to add some changes, new features to your product. You need to track each change, and version control systems like Git enable this for you. Git also enables, or version control also enables effective collaboration, an easy way to find where an error was introduced and revert. And there are, of course, many platforms that you can use, like GitHub, GitLab, and Bitbucket. Employ code reviews. So every change that you try to add to your software, make sure there is a second pair of eyes on it. Basically, somebody should review your code. This can catch bugs early, and you actually share the knowledge in your team through this process. Test your code, write unit and integration tests for everything that you implement. This, first of all, ensures that your code is correct, that works correctly, that does what it's intended to. And then you're more comfortable into extending your your product with new features, refactoring it to make it work faster, or run on another platform. Try to write modular code. That means that every time you write a function, try to make it that it does one thing, basically, and does it well. Then that makes it more easy to be reused in other features in the product, and it makes it testable, understandable. When you see a small function, it's quite well named. Also, it's very easy to understand what it does, but also document your code. So describe what it is the intent between any piece of code that you write. This, of course, being part of being compliant with these standards that I mentioned, also helps you helps you onboard new team members, and describes how to properly use and maintain the product. Enforce coding standards, so try to keep some rules on how you write the code to make it more readable, but do this automatically through using some linters, for instance. And last but not least, implement continuous integration and deployment. So automate all of this, and with each change that you do to your software, make sure you run all the tests to make sure that the product is in a working state, and so on. Yes, you do this when you write software, but when you develop machine learning models, everything what I said about software development applies, because to train your models, to do inference afterwards, you implement some kind of software. The best practices, besides the same best practices that apply for software development, involve then also model versioning and dataset versioning and MLOps. So what's that? Model versioning is basically tracking all the different models that you develop. So you do a lot of iterations when you try to solve a problem with machine learning. You try different features, different parameter configurations, and so on. If you keep track of all these changes, you can see how your model has evolved during development. And this is another part that you need to be able to document to comply with the standards mentioned previously. It will enable for your work to be reproducible, easily revert back to previous models in case some new change actually destroys the performance of your model, for instance. And enables traceability. So every change, you know where it came from, who did it in the team, and so on. How to do this? Often in machine learning, you will do the data preprocessing or the training, hyperparameter optimization, maybe in a Jupyter notebook, for instance. Notebooks are great for experimentation, but when you can, you should also version this code. So extract it from the notebooks and write proper libraries or modules. Then each time you create a new model, save the model and the environment parameters and so on. Am I out of time, or? 1 more minute. Okay, I'll try to speed this up. So I separated dataset versioning as a separate point because it's also very important. Like I said at the beginning, it's very important that your data is correct and up to date, and so on. And you can automate all this with MLOps practices or CI/CD for machine learning. Some tools that help you do this. I will not get into much of those. And some final thoughts. If you follow these best practices and combine them with an issue tracking system such as GitHub issues or Jira and Confluence, document your software requirements, and write a few technical files describing the product and the relevant procedures, this will actually create a QMS that is sufficient for regulatory approval. Additionally, please consider model interpretability and explainability. I take here an excerpt of the summary of the EU Act, EU AI Act, which I think you're going to hear soon more about, and integrate these explainability methods into your pipeline. This is not easy to do. You need to find the balance to actually do the research, so do fast prototyping, which you usually do in academia, and the stricter operating procedures in industry. Find the balance to actually develop your product, and, but at some point, please implement QMS. Yeah, and no time for questions. Sorry about that. That's it.
Thank you, Boris. During the coffee break, maybe people can get some industry secrets from you.
Please come and ask.